I'm Vu To. I'm taking a PhD in chemistry program in University of Manitoba since 2011. I did my undergrad study in Vietnam and worked for 2 years before coming here for graduate study. As to my research, my group focuses on studying protein structure and dynamics by NMR. Protein are one of the most abundant and important molecules in the living body. They serve a wide range of functions from giving structure supports to cells to helping immune system to fight off disease. Protein malfunctioning leads to serious diseases, for example, diabetes due to insulin deficiency, or Parkinson, Alzheimer, Huntington diseases due to protein misfolding. Many pathogens use proteins as their weapon when infecting a host body, and some other proteins called prions are pathogens themselves, such as mad cow disease, as you may know. It is widely recognized that representing a protein as a single static conformation is inadequate. Many protein biological functions are expressed via dynamics, and to understanding structure and dynamics, it's important to understanding functions, predicting and engineering protein behavior, and designing drug targeting proteins to kill diseases. For the last few decades, scientists are more and more aware of intrinsically disordered protein, a group of proteins that doesn't have secondary structure and are completely flexible. However, proteins in this group are found to bind to different targets, expressing different functions. This is explained by their high dynamic property, which allows them to sample different conformations upon binding to partners. The protein of our research, HIV-1 transactivator of transcription, belongs to this intrinsically disordered protein group, and as you may know, HIV-1 is a retrovirus suppressing the immune systems and causing death to millions of infected people. HIV-1 transactivator of transcription protein enhances the efficiency of viral transcription, and without it, the virus cannot replicate. It is also found to bind to a number of other targets in human cells, causing different AIDS-associated symptoms, one of which is the well-known HIV dementia. TAT, similar to 14 other HIV-1 proteins, is a potential target for drugs that cure HIV. Our group is interested in studying the dynamics and structural changes of TAT upon binding to different human proteins. Since 1984, when the first protein structure was determined using NMR, NMR has been considered one of the most important methods for protein structure determination. Along with X-ray crystallography, NMR can provide information about the protein in atomic level of resolution. However, the protein of interest, TAT, can't be crystallized because it is a disordered protein, so NMR is the method of choice. The protein of our research is isotopically expressed using bacterial system. Protein plasmid is bacterial codon optimized and transformed into E. coli competent cells. Transformed cells are grown in media supplemented with isotope nutrition source of carbon-13 and 15 isotope labeled amino acid or precursors that suit the requirement of NMR experiment. After expression of protein, the cells are harvested by centrifugation. Labeled protein is then purified by gravity affinity column and further purified by FPLC if needed. NMR experiments on the purified sample are done at University of Manitoba NMR facility. Acquired data is processed, visualized, and analyzed to assign chemical shifts of protein residues. Calculated ratios are also used as input for the structure calculation process. NMR relaxation and hydrogen exchange experiments are done to acquire dynamics information. Hopefully, my research will deepen our understanding of this viral protein and contribute to a better understanding of HIV infection.